Okay, we're going to start part 3 and final section of chapter 3, all about the cell. This part of the chapter is more about how we make more cells, so the process of cell division and what has to go on in there. When we talk about the cell itself, there is something, again, there's always terms with everything we do, but there's something called the cell cycle. And the cell cycle is basically like a clock. It's the time that the time that the cell spends in each stage of its cycle, and it goes around, like from, if this were noon, it goes around the clock and comes back and then starts over again. So it's divided up into these different stages, and you can see the stages on here. Part of those stages are what we call interphase, and interphase is just the part where the cell is doing whatever its job it's supposed to do. So it's getting nutrients, it's growing, but importantly, if it's supposed to be making something like an enzyme to digest your food, your carbohydrates or something, it's doing it at that time, okay? So it's doing the necessary function. Then it will move into, after the cell has grown to a certain point, it's going to move into a stage that we call mitosis. And you can see that each of these, like interphase, is from here, this yellow, all the way through the brown. So it's actually this whole 3 stages right in there. So we've got these substages in there. Mitosis is the same way. Mitosis is actually where we're going to have cell division occur. So this is where the cell is going to divide, the nucleus is going to divide, and all the nuclear material are going to divide. And again, there's these substages, and it's all of this green in here, plus this little one called cytokinesis. That cytokinesis is at the very end of cell division, and it's where after the nucleus has divided, the cell is the rest of the cell has to divide too. So let's start with the first stage, interphase. Interphase, like I said, is the time where the cell is doing its job, but it's also getting bigger. It's come out of a previous division. It was smaller at that time where the cell had taken and it had divided in half, so it's now a small cell, and it's going to start growing and getting larger and larger and larger while it's performing the function that it needs to keep your cell and your body and your tissues and everything alive. At some point during interphase, so here's G1 right here, this is the first stage, it's progressing through this phase and it's getting bigger, doing its job, like I said. At some point it gets to the point where it's pretty large, and there's these little cell signals that say, hey, the cell is going to have to divide somewhere in the near future. We're getting too big, we're getting inefficient, can't get enough nutrients in to feed everything inside the cell, can't get enough waste products out, whatever. I'm going to have to divide. So it's going to enter a phase where it has to build the things that actually will be used in each cell. So it has to make a copy of the DNA. That's one of the steps. Make a copy of the DNA. And it's going to have to build more of those organelles that we talked about, because each new cell that comes from that division is going to have a full set of mitochondria and Golgi apparatus and whatever else that's in there, ER and that kind of stuff. So it's going to have a set of DNA for each cell, and it's going to have organelles for each cell. So it does do this prior to division to get ready for that splitting. And then that's S and G2, and then it will enter the point where it's going to divide. Notice how small the division stage is. Division stage is this small little piece of the pie right here. The cell's not doing what it's supposed to when it's dividing. All it's doing is dividing. So you want it to kind of go, okay, divide, get back to job. So, take your rest, basically, divide, get smaller. Now it's time to go back to work. Let's get back to the point of what you're here for. Okay, so the subdivisions of interphase then. So here's this, in this one we're talking about right here. This is the start. So this is the start of interphase. This is all interphase right here. And it's going to go around. It's going to go to through G1 phase. It's going to go through S phase, and it's going to go through G2 phase. So this right here is the end of interphase. So interphase is everything except this one little small section I left out right here. So the first one is called G1, and really the G just stands for gap. We weren't real creative with the name. But the gap 1 phase, or G1 phase, The cell has just divided, so it's small. It needs to recover from that division, so there's a little recovery from division. Importantly, there's a lot of growth going on, and very importantly, it gets back to its job. Whatever it's supposed to be doing, it starts doing it again. So your cell starts doing this. All the while it's progressing through this, it's getting larger and larger and larger. When it reaches near the end of G1 down here, has to decide, are we going to divide because we've gotten so large, or does something else need to happen? And that the reason it has to go through those, and it's not like there's a brain that divides that. There's chemicals and everything that decides this process. But you really don't want to divide a cell if something had gone wrong in mitosis and this cell isn't doing its job correctly. You kind of would want to end that process right there and not divide that cell and have more cells doing the wrong thing, right? So there is this, what we call a checkpoint right here that says, do we want to leave and just never divide again? Do we want to kill the cell off and then never divide again? Or do we want to continue into S phase? Most cells are going to continue into S phase because they're going to need to divide and go on. But if there's something wrong, something that needs to be corrected, or some other reason we're going to talk about a little bit later, it may just stop there and not go any further. When it enters S phase, S stands for synthesis, and synthesis means to make. So what's happening in S phase is the cell is making DNA, because each new cell is going to have a full set of DNA. I can't take my DNA if there's 5 chromosomes, and I can't split those 5 chromosomes between 2 cells because one of those would be broken, right? So you got to have a copy made of each one of those chromosomes. So if this is one chromosome, this is one, and so on. I got to make an exact copy of each chromosome so that the new cells have an exact copy. And then I can split that, and each new cell has its full set, right? Okay, so that's what's going on in there. So S phase, you have this replication, the synthesis of new DNA, so that I can give a set to each new resulting cell at the end. Again, cell still doing its job. So don't forget that that's going on during this point here. These are just like new things that are going on. It's always doing its job during interphase. And then the final stage of interphase is gap 2 or G2 phase. During G2 phase, it's really preparing for that upcoming cell division. Remember, it's still doing its job. So that's going on too. But now it's going to make some organelles. It's going to make the apparatus to divide. It's going to start putting things together to say, hey, final prep, let's go to division. All right. So cell division is really divided into 2 things. We have nuclear division and we have the rest of division. So the nucleus contains the DNA, it contains the chromosomes. You got to split that. It's really important material because that material is the information for the cell to make proteins and to go through everything it needs to. So we got to really concentrate on that. And that's a specific part of this cellular division or this nuclear division. We call that karyokinesis. Okay. The rest of the cell dividing, splitting up the cytoplasm, splitting the membrane in half, to pinching it so that each cell gets some, dividing out the organelles. That's what we call cytokinesis. So that's just like everything else. If it's not nucleus, it's part of cytokinesis, all right? But what we start with is one cell, and what we end with is, right down here, 2 identical daughter cells. Identical in the fact that their DNA is going to be exactly alike. When we made a copy of the DNA during S phase here, it was meant to be identical DNA to be split between those 2 cells, okay? So then we're going to form those 2 identical daughter nuclei, which then eventually will become 2 identical daughter cells when you split the rest of the cell. There's 4 stages of this nuclear division, mitosis. So mitosis really refers to the nucleus dividing, that karyokinesis part of it. Shorthand is PMAT or PMAT, and it stands for each of the stages. The first stage, P, is prophase. During prophase, our chromatin shortens, thickens, and becomes chromosome. So let's step back for 1 second and talk about these terms. And I talked about it earlier, but let's refresh those terms. So here's my cell. I'm going to draw a really big nucleus in here. In the nucleus, I have genetic material. That's DNA wrapped around chromosomes or wrapped around histones. and we call that chromatin. Remember I said when it's long and thin, looks like a bowl of spaghetti, right? Because you can't pick out one chromosome from another. That's when we refer to it as chromatin. So it's really just a form of what DNA is in. And that's during interphase. So it looks like this during interphase when it's doing its job. During mitosis, it's really kind of hard to go in here and say, here's one chromosome, here's another chromosome, here's another one. Let's split that up. If I start to take this little bowl of spaghetti and split it up, what I'm going to do is probably break some of those noodles, right? Or break some of that DNA. Not good for your cell. So before this long, thin, chromosome can be divided because it's all mixed up with the others, it goes through a series of actions where it starts to wind up. Okay, and remember it's doubled, by the way, because in S phase it doubled, so you really have these two long strands hooked together, and it starts to wind up. Okay, so it's winding up and curling up and curling up, and it becomes much smaller. And each half here is going to wind up and become much smaller. And what you end up with is this, if you can follow the spirals down, is what looks like an X. It's much smaller than it was before. It's much more organized than it was before. And it's a neat little package. Now I can take that package, instead of this whole big bowl of spaghetti and try and divide them out, I can take these individual little packages and easily divide them out. When the chromatin has shortened and thickened and become this stage, now we refer to it as a chromosome, okay? It's the same material. It's the exact same thing as you found in the nucleus when it was chromatin. It's just in a different form, okay? It's shortened, it's thickened, it's curled up, and then we look at it and it looks like an X, okay? So those are terms you really do need to get to know. Chromatin, long and thin, during interphase. Chromosomes, the X's that are wound up, short and thickened little packages for cell division. And this is really only during mitosis that this occurs. When we see pictures of DNA and we see maps that show where genes are, they always show it during this phase because we force the DNA into this stage. It's just much easier to see and map out if it's a small little package than if it's this long wound mixed up bowl of spaghetti, right? So we tend to force it into that sort of outcome there. All right, let's erase all this. All right, so during prophase then, we're going to begin this cell division. So that is the point where we need to shorten and thicken that chromatin and force it, and the cells force it into that chromosome conformation. So it's the same thing, it's just now a different shape. So now all my little DNA are in these little X's called chromosomes. Now I've got a package I can divide pretty easily. Since all the DNA is wrapped in a nucleus, the nucleus is going to be hard to move DNA to two different ends of the cell if it's inside sort of its little room, its little compartment. So that disappears. So the nuclear envelope disappears so that now I can divide all these chromosomes out into two different cells, split them up into two parts of the cell so that we can divide. So nuclear envelope disappears. The nucleolus is another thing that disappears. Remember, the nucleolus is that dense region of RNA that makes ribosomes. It's kind of in the way right now, so we just kind of disperse it, get rid of it. It'll come back later. All right. And then these things called centrioles move to the end of the cell. And again, we talked about centrioles. So if this is my cell, I'm going to have my chromosomes in the middle, where the nucleus was. That little nuclear envelope was here, and it's disappearing. And then those little centrioles move to two ends of the cell. They look like little T's. They're going to be the organizing units that actually allow for those chromosomes to move. So they're gonna determine where those chromosomes go. Okay, so they have to move from where they were located, sort of centralized. They move to two ends to start that dividing process, that moving apart process. Centrioles, by the way, are only found in animal cells, so you have them in your cells. And then these things called spindle fibers start to radiate out from these centrioles. So they're right there, and they're going to attach to, I'm trying to erase that. Here's my cell. They're going to attach to each chromosome, okay? So they come out from those centrioles, and from each side they attach to the chromosome. So really, for my chromosome, I'm going to have a spindle fiber from each end. Here's my little centriole. So this centriole sends out a spindle fiber to the chromosome right there, and this one sends it out here. Okay, so let's talk about that for a second. This is here's my chromosome, right? Looks like this. This portion right here, so the whole thing's a chromosome. This portion right here where those spindle fibers attach, that's called a centromere. Okay, so not to be confused with centrosome. Centrosome was the centrioles together. So I have centrioles, centrosome. These are centromeres. The ends of my chromosome, right here, 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 and here, those are called telomeres. So that's kind of your anatomy of a chromosome real quick. So I have telomeres on the end, but I have the centromere. And the centromere is a bunch of protein and different things, and that's where those spindle fibers will attach. What's going to happen is by attaching there, it's going to allow these chromosomes, that X, to be divided right down the middle. Half of that X will go that way to that centriole, and the other one will go the other way to the other centriole. So my X divides into two halves, really, eventually. Okay, just splits right there. That's that centromere. All right, next stage, metaphase. With metaphase, here's my cell, right? I don't have a nucleus anymore. Prophase, I had all these chromosomes still centralized in the center, but in metaphase, they start to be pulled towards a line. So we have them in a line down what's called the metaphase plate. So that imaginary line down the center is the metaphase plate. Remember, I have centrioles on either side. Each of those centrioles send out spindle fibers that are attached to those chromosomes, and they're organizing that. So those spindle fibers and the centrioles are what's organizing that lining up of those chromosomes. And the reason it does it is basically an inventory. Do we have everybody replicated? Do we have each of the chromosomes present? Let's put them in order so that when we split, and this side becomes a cell and this side becomes a cell, each cell gets an exact identical half. You don't want this sort of random movement of chromosomes. Some of you go here and some of you go here, because then those cells won't be identical. We want exact matches to split, and half go to one cell and half of that exact match go to the other cell, so that the two ending cells at the end, after this is all done, are exactly identical to each other. So that lining up is very quick, but it's taking inventory of what's genetic material there is. And when you look at it in a picture, so here's a cell, right? Here's in purple, those are the chromosomes. I can see very light pink, I can see the spindle fibers, and where these come together, those are the centrioles. So I can see the centrioles where all the spindle fibers come together. So this is one cell. Those chromosomes are lined up down the center of the cell, okay, like that. And you can really distinctly see it. When I look at this cell down here, right, this one's in interphase. I can't really pick out chromosomes. I can see a dense region. This one's a good one over here. See that cell membrane around the outside? It's a better one. Let's talk about that one instead. So cell membrane around the outside in purple, but you can't really see the chromosomes in there. I can tell that's interphase because I can't pick out any chromosomes. Whereas in this one, when you look here, they're very dense, they're darker in color, and I can actually see almost little X's in there, okay? I mean, almost. Have to really look. But metaphase, they're lined up. Once you've taken inventory, once it's lined up, it's going to move to the next phase. And the next phase, PMAT, is the A. So that's anaphase. So anaphase, where they were lined up right here down the center, now those X's have divided. Half of that X, it actually pulls it opposite, so we're going to draw it opposite. Half of that X goes this way, and half of that X goes this way. So the X's that were together like this flip, so they just kind of flipped out. So the center point of that X is opposite direction. And they're being pulled to those centrioles by those spindle fibers. okay? And then that goes for every single chromosome in there. So they've left the center and they're being pulled apart. So what you see here, not in drawing, is these two little groups of chromosomes being split apart. But notice it fans out. So really pay attention to that fanning out. You're going to need to know this for identification for both the lecture test and the all of these stages for the lecture test and the lab test. So really pay attention to these kinds of characteristics. So see how it fans out and it kind of comes to a point, but you have two sections here. That's anaphase. So anaphase, those chromosomes split, okay? The spindle fibers split them apart and they start moving to the opposite ends of the poles, right? They're not really all there yet. They're being pulled apart. So it kind of looks like this pulled like this. Okay, and then we come to the last stage of mitosis, not the last stage of cell division, but the last stage of nuclear division. So in mitosis, notice where I had those chromosomes being pulled apart, but they look fan-shaped in anaphase. Here, they're starting to circle up again, okay? So we're starting to form two new nuclei. So that means the nuclear envelope reappears. The nucleolus, that dense region of RNA, reforms, okay, in there, so it can go back to work and make some more ribosomes for protein in a little bit after the cell gets back to normal. And those chromosomes relax. They go back to being the long spaghetti-like chromatin form of DNA in there. At the same time that this is happening, so this is telophase, where we're talking about forming the new nuclei. The spindle fibers start to go away too. You can still see some of them attached through the center, but it's going away. But at the very same time, notice this cell, here's the outside of the cell. Notice it starts, and these arrows are here, it starts to pinch in, okay? So at the same time that telophase is occurring, the last part of cell division is also occurring, and that is cytokinesis. So cytokinesis is when the rest of the cell divides. Kinesis. So the cell membrane splits, right? The cytoplasm splits, and the organelles are divided out. So that's happening at the same time that telophase is. But remember that they're two separate sections. Telophase just refers to what's happening in the nucleus. Cytokinesis refers to everything else outside of the nucleus, this splitting right here. But they do occur at the same time, start to occur. So here's kind of a textbook view of this. This is the first part right in here. So we start entering prophase right here. Nucleus divides. Here's the centrioles in purple. They move to 2N, so this is later prophase. Notice the nucleus, they've got it broken up more. Chromatin, that was here in the first step, is become chromosomes at this point, and the centrioles are moving apart. Spindle fibers are starting to form. So this is later prophase right here. So this is like interphase. 2 is early prophase. 3 is later prophase. They're starting to line up. 4 right here, this is actually metaphase. Okay, these are both prophase. Metaphase, and then you have anaphase right here, and then we have telophase right here. And then see this forming here, this is cytokinesis is finishing up with the end of telophase. And then we're back to interphase with 2 new cells. And these two cells here, called daughter cells, are exactly identical to each other, and they're exactly identical to the cell that they started out as. All right, so cytokinesis, remember, is that last stage of everything else dividing, and it happens really at the same time as telophase does, but the cytoplasm divides. This division between the cells, like a meat cleaver cleaves things in half. It's called a cleavage furrow because it starts to divide. The membrane just pinches in, and then it eventually pinches completely in half, and you have 2 cells. Organelles divide between the 2 daughter cells, and now we have 2 identical daughter cells. You started out with one parent cell. We have two identical daughter cells at the end. So what regulates the cell division? There's a lot of things that regulate it. Some cells divide all the time. When you look at your skin comes in contact with all kinds of surfaces, really gets a lot of abuse, even wind and rain and clothing and whatever else, not just you knocking it and hurting it. It really gets a lot of abuse, and it's there to protect you. So you're constantly knocking off cells, so you have to replace those cells all the time, right? Your blood cells, especially those red blood cells, don't even have nuclei, so they can't go through mitosis actually, but they have to be replaced in the blood and bone marrow. So you're constantly, things that get worn out, that get used up very quickly, we replace them very often and very quickly. Other cells, like your liver cells, they'll divide and then we say rest, but they don't really rest. They just sit and do their job for a very, very, very long time. And then maybe they get old, they get damaged, and it's like, oh, we need to make some more. So they'll enter back into that cell cycle and start going through division. So they go through this what we call a GO phase, which is a rest. So if this is my cell cycle going around, this is mitosis right here, portion of it, and this is interphase. So G1 is first. Before it goes into S phase, remember that marker I said, that checkpoint? Sometimes the cells will enter out and go into a GO phase. And for liver cells, that's kind of temporary. It's like, we can stay here for a while. We don't need to make more cells. And then maybe you need to make more cells, so it will enter back into the cell cycle, go through S, make some, and then leave again and go for rest. So they don't really make copies very often. Some cells, like your neurons that I have listed here, once they differentiate, so as a fetus and as an embryo and fetus and all that, and you're differentiating and you're making neurons, they go through the division of making a cell, and then they specialize into neuron, and then they just stop. They go into GO phase, and they never come back again. So that means you never, ever actually make any more once they've differentiated. They stay in GO forever until you die. So damaging your neurons can be a problem because you can't replace them, right? So it just depends on the cell as to how fast it goes through cell division. Some things often and quickly, some things not at all, like the neurons, and then some things there's a in between the two. Don't need to make them as quickly as the skin, but I do need to make some more every now and then. All right, so remember my chromosome I showed you? So here's the X of the chromosome. This is in the center, that's your centromere. These ends that are in red are called telomeres. These telomeres play a part in cell division. There's many parts in cell division and why things divide and when they divide and aging of cells and your life and everything. Telomeres play this part. Telomeres are these ends of the chromosomes. Every time your cell divides, you have to make a copy of your chromosome. So if this was the original half, it made a copy over here. And then that's what's going to be divided during cell division. Each half will go to a new cell. Well, these things get old after a while. Every time that you divide, you lose actually a little bit of the end of the chromosome. That could be really bad if it's important genetic material, but the telomeres aren't. The telomeres are these just repeating sections of DNA that really don't code for anything. So you can afford to kind of pair this off and cut this off at the end during every cell division. But unfortunately, at some point, you are actually going to enter into like this purple section that codes and has genes. That says, hey, we're done. We can't keep making a copy of something and destroying the genes. So every time it shortens, it kind of shortens the life of that chromosome and the lifespan of the cells themselves. So they can go through a limited number of divisions, and then eventually they're just done. The cell's done, it's old, it gets destroyed, and it doesn't go through the cell cycle anymore. And that's a part of the aging process. It's part of the cell aging process, and it becomes part of our tissue aging process and our process as an animal and as a human being. Imagine if you could repair all these. You'd never age, right? That's theory. There's a lot of research out there. Some organisms and some cells can do that. Our normal body cells always age. Our gametes, which are our sex cells, you don't really want to pass off shortened chromosomes to your offspring every time you make an egg or a sperm, right? So we actually do repair them for that process. So we have the ability to repair those and keep them from from wearing off. But it's only for certain purposes. Not all of our cells know how to do that. Another thing, unfortunately, is cancer has learned how to repair them. So cancers are called immortal cells because they never age out and they never die until, of course, their host pretty much dies. All right, another thing is the size of the cell. So division of the cells are, remember I told you that during interphase, the cells start out small and they start to grow, and at some point they really become inefficient. They cannot get enough food coming in, and enough waste products out to really allow for the interior of the cell to get enough food and to get rid of waste products efficiently. So the larger the cell gets, like this big cube right here, we're just going to make it a cube so it's easy for math and to be representative of a cell. When it gets too large here, it really gets inefficient, can't get enough food. So it's really much better to be a small cell. But the cell's going to keep growing. So as it's going through the stages of mitosis, of interphase G1, it's getting bigger and bigger. And then at some point, inefficient, let's divide down and get small and go back to being small again. Okay. So here's the math that's behind that. If you think about surface area and for volume, this light purple, each of these sections here, light purple here, this is what we call surface area. So it's one surface of a cube, and every cube actually has 4 surfaces. So all of this is here. So if I look at it, it's got a surface on the top, it's got one on the bottom, it's got some on the back sides that you can see kind of proportion of here and here, and it's got it all around. So it's got 1, 2, 3, 4, and then one top and one bottom. So it has 6 surfaces, right? So every cell has 6 surfaces. Volume is defined by what is on the inside. So if I went inside of this, what is inside that cell? That's its volume. So I have to look at the surface to its volume. How much surface, because the surface is the cell membrane, right? And the cell membrane is the only place things can go in and out of the cell. So that determines the rate of moving stuff in, my surface area, my cell membrane. The volume is what needs food. It's got organelles, it's got cytoplasm, it's got the nucleus, it's got everything that needs food and things to build things and everything else. All right. So when it gets too large, it's inefficient. So what we do is we compare these two things. Let's compare the surface area to the volume, and we call that surface area to volume ratio. Those two little dots tell you it's a ratio. I'm going to compare the two. My little cell down here is a 1 millimeter cube. So it's 1 millimeter on every single side that I go to, all right? So when I calculate the surface area for it, since there's 6 sides, it's 6 times length times width. So I take the length and the width, and since it's all 1, it's a cube, it's 1, it's going to be 6 times 1 times 1, and that equals 6 times 1 is is 6. Times 1 is 6. So my surface area is 6 square millimeters, okay? Since we had millimeters down here. The volume of that little cube, volume is length times width times height. So my volume then is 1 times 1 times 1. 1 times 1 is 1, 1 times 1 is 1, so we're at 1 millimeter, cubic millimeters. So here's my surface area, here's my volume. I have a 6-to-1 ratio, surface area to volume. They've got it with a slash, that's another way to write it. So you can write it with the 2 dots, or you can write it like a fraction, all right? But it's a 6-to-1 ratio, meaning that there's 6 times more surface area than volume. It's quite a bit of surface area. All right. When I move up to this last cube over here, that's a 4 by 4 by 4 cube. So all sides are 4, right? It doesn't matter which way I go on it. My surface area for this one is going to be 6 times 4 times 4, right? 6 times 4 is 24. 24 times 4 is 96, right? Wait, 24 times 48. Yes, 96. I did that math right. Sorry. Had a moment there. 96, right? So it got bigger, went from 6 to 96. Our surface area definitely got bigger, right? Volume is 4 times 4 times 4, so it's length times width times height. Volume is 4 times 4 is 16, 16 times 4 is 64. So my volume got larger too, where it was 1 before, it's now 64. This thing got significantly larger. But we're going to have to compare these two numbers to these two numbers right here. So we're going to have to reduce. And what that ends up being is when I reduce and do the math, 64 and a 96, what I end up with is a 1.5 to 1 ratio. Yes, my surface area is still larger than my volume. It's 1.5 times larger than the volume. But look at it here. Even though it's larger, it's much less of a ratio. My ratio just got smaller. This one here is 6 to 1 ratio. This is a 1.5 to 1 ratio. So this has the surface area to volume ratio has gotten smaller, which means its efficiency has just been reduced. So the smaller cell actually has a higher ratio and higher efficiency. So a smaller cell, higher ratio, higher efficiency. Larger cell, smaller ratio, smaller efficiency, less efficient. If you want to think of and try and put this in terms that you can kind of realize what's going on here. If you have $10 in your wallet right now, would you rather have 6 times the amount of that money or 1.5 times the amount of that money? I'd like to have both. But if I were given a choice, I'd rather have $60 than 15, is what that's saying. So 1.5 times would be just $15 in my wallet versus 6 times 10 is 60. So this is the preferable situation to be in. So once the cells get too large, they go through cell division so they can get small again. Okay? So cell division is regulated by size, is one of the things. So size, that's one of them. So we had telomeres regulate sort of the lifespan of the cell. Size is going to regulate what's going on with the size of the cell. And chemicals. You have hormones and growth factors that are going to be regulating when and how often a cell goes through cell division. All right. So some hormones like growth hormone. You also have hormones and growth factors like mitosis promoting factors and different things that regulate the cell itself or growth of a bunch of cells at one time, like in growth of a human body. So you have to keep in mind that these are in here. So growth hormones, growth hormone, GH is one. Growth factors like mitosis promoting factors. Another one is something called CDK, and it refers to the specific type of chemical that's in there that's going to regulate this. But those are different things that are going to regulate whether the cell divides or not. Okay. And then you have skin growth factors. So epidermal growth factors stimulate the growth of new cells constantly. So we had telomeres regulate how long, how many times a cell can divide. You have size of the cell that regulates the mitosis. You have hormones and other chemicals, growth factors that regulate how many times or when a cell will divide. And then we have this one called contact inhibition. So this is another one. Contact inhibition just talks about the contact that the cell comes into with other cells. So in this first Petri dish right here, you see these cells. I have no idea why they have little flagella on it, because they kind of look like skin cells in the last one. So I'm not really sure what's going on in this picture, but it's the picture I took. So that's the one we got. So here, this cell is independent from this cell, and they're not in contact with each other. There's a stimulation for them to start going through mitosis if they don't touch another cell, okay? But after a while, when you get to this last Petri dish, each cell is in contact or touching another cell. That says stop growing, all right? Stop growing. We don't want to continue growing because at some point you would just bulge up and it would be bad for the cells. So as long as they're in contact, they're not going to go through rapid mitosis. When they don't have contact with each other, they will go through rapid mitosis. Here's the reason. If I take my arm and I get a cut on my arm, those two sides of the cells are no longer in contact with each other. That's a wound. That's dangerous. I need to have some sort of stimulation that says, let's seal that up very quickly and get cells to replace that section in there so that you don't have this big gap. So when they're not in contact, wound, right? They're going to want to be in contact. So they go through a more rapid mitotic division so that they do come in contact with each other. And then they'll slow down again and go back to their normal pace. All right. Unfortunately for us, some cells lose that ability to recognize the touching factor. Okay. They lose that ability to recognize contact inhibition. We call these tumors. Tumor cells, once they touch each other, they don't stop. They continue to divide, and they do build up. And that's why we can feel them. So if you're checking for breast cancer or you're checking for skin cancer, you can see a rapid growth in an area of cells. You can feel a lump because they don't stop when they come in contact like other cells do. So that's one of those things that causes them to be dangerous, because tumor can put pressure on organs. These cells can sometimes break off and move to other portions of the body and do the same thing in other portions of the body. Tumor by itself is not necessarily dangerous unless it's putting pressure on something. A tumor that is spreading to other regions is dangerous, and one that is putting pressure on maybe your trachea or your brain or your heart or your lungs or something, that would be dangerous. So we divide these tumors into two types. Benign is the type that's not spreading. It's just in one area and you can feel it. And sometimes you can just have that removed. So like a fatty tumor maybe that's under the skin, you can have it removed. Or if a tumor is detected maybe in a lung or something, they can remove that section of cells. As long as it isn't actively spreading to other cells, it's just localized in that region. We remove it, it's gone. But if it's categorized as malignant, mal means bad, that is invading other cells. So those little tumor cells are actually leaving and going to other parts of your body, and they can do so through your lymphatic system or through your circulatory system and set up residence somewhere else and then go through rapid division themselves in other areas. So now I'm going to have tumors not in one area, but all over the body as they move. That's called metastasis. So these cells are metastasizing, and that's when we refer to it as cancerous. They're invading other cells, they're rapidly growing, and they're causing all parts of the body to become dangerous. You can see a little video link up here. That video link is in D2L for you, so you can go look at this. It shows metastasizing cancerous cells, and it shows how they break off and how they move. It's a little animation. It's short. It's good. Okay, and then we have these things called genes that also regulate cell division. So we're going to have genes that regulate cell division. Normally, these genes would either, it depends on the type. Some are not active. Some are active because they're constantly patrolling for cells that aren't good. Okay, so we have these two categories. Onco means cancer, and oncogenes are genes that will increase cell division. Normally, these genes are in the off position, so we have them turned off in our cells. But if they get a mutation and they turn on, and both sets of genes turn on, then those cells will start increasing cell division, and that's become tumorous. Okay. In most cases, most of your cells, since we have two copies of every gene, you have two parents, so you got a copy of every gene from both parents, one from each parent. So if I have a gene from my mom and I got a gene from my dad, and they're both in the off position, right, like they should be, so they're both, I'm going to put X's for off, both off, I would have to turn both of them on, so go from an off to an on position for that particular cell to become cancerous. Sometimes we say somebody is predisposed to cancer, and that is because here's my first gene, and from mom or dad or whatever, here's my second gene. And one of them I inherited in its normal off position, but one of them I inherited in the on position. So it was already mutated when I got it as a zygote, as an embryo and a fetus, right? So it was already on. So really, this one's already on. All I have to do is get one mutation, a mutation on that one, turn that one on, and now that cell will become cancer. So I'm more likely to have cells that become cancerous. Doesn't mean that this will happen. It's, it may never happen. This may always stay off and you're good, but you're just more likely than somebody who inherited two off genes to get that. Okay. Other genes are what we call tumor suppressor genes. We inherit these genes normally in the on position. So these are always on. And we want them to be on because their job is to make a protein that goes around and checks the cells to see if the cells are bad. So here's my little brown bad cell, right? So they're making proteins that are constantly patrolling for something that's wrong. And when they detect it, they're going to destroy that cell so that it can't make more copies of itself, and it can't make some more. So these cells we want in the on position so that they can do their job to patrol, right? Well, what happens if these go to the off position? So my on just got turned off. If one gets turned off and I still have one on in that cell, we're still good. But if they both get a mutation and they both turn off, right, then I no longer have a patrol. I no longer have the ability to check for the cell the incorrect part of that cell to turn it off and to kill this cell off. These guys are what we call immortal cells. These are cancer cells. So really, in order to get cancer, and once again, you could also inherit, by the way, one off like it's supposed to be and one on. So that would make you more likely to become cancerous if it was already in the off position. But for a cell to become cancerous, they're going to be doing this. Things go wrong. You get mutations all the time. But really, I have to have this occur. I have to have my tumor suppressor genes that are supposed to be on turn off in the cell. So both of those turn off. And I have to have these oncogenes that were supposed to be off both turn on, right? For this cell to become cancerous. So both of this has to occur in the same cell, right? So I have to have 1, 2, 3, 4 little mutations occur, unless I inherited some, for a cell actually to become cancerous. So it's not likely that that will ever happen. Otherwise, we would all be running around with a bunch of cancers. And most of the time, anything that goes wrong gets checked and the cell gets destroyed. So you really have to have all of that occur. This is what we call the multiple hit hypothesis. It's not just one thing that goes wrong with the cell for it to become cancerous. You actually have to have multiple things go wrong with the cell before you can have it. Now, if you inherit an oncogene already from one parent or another, you really just have to have one more mutation. But still, you have to have one more mutation that randomly occurred in that cell for it to actually occur. So for the one oncogene thing, that would be a 2-hit hypothesis. But remember, we also have that other set, the tumor suppressor genes that also have to go wrong too. All right? Multiple hit hypothesis. So we really do, every time something goes wrong, and it goes wrong, with a part here and there, it's checked, there's balances, we destroy the cell, and we go on with it. It's when that one happens that doesn't get found by the cells because you've had so many mutations occur. Things like, for skin cancer, things like too much sunlight and stuff like that causes more likely for skin cancer to occur. Smoking increases your chances for things to go wrong in those cells. So you can accumulate those over a period of time, and you're much more likely to see cancer occur because you're forcing a situation where those cells are going to get those multiple hits. All right, stem cells real quick. Stem cells are cells that we all start out with, and we all have, and we still have throughout our life. These are cells that are going to grow into any type of cell. So when you first are become into being, right, you're an egg, and a sperm comes in, the nucleus of the sperm enters a cell, and you get one set of DNA from mom, full set, 23 chromosomes, and one full set of DNA, 23 chromosomes from dad. So now you have what humans are supposed to have, 46 chromosomes. One copy from mom, one copy from dad, one copy from mom, one copy for dad, for every pair of chromosomes. So humans have 23 pairs of chromosomes because they got one set of each pair from each parent, with a total of 46. Okay? You are initially just a ball of cells, one single cell with 46 chromosomes in it. Immediately, that cell, which is now called a zygote, by the way, a fertilized egg cell is called a zygote. That zygote starts to divide, and it divides into 2 cells, and then divides into 4 cells, and so on. And within a couple days, you're this big ball of cells. Okay, we call a blastula. So you're in the blastula stage, and it's this big sphere of cells. Any of those cells could become any part of your body, right? It's going to start, that big ball of cells is going to start, when there's enough of them, to send messages that say differentiate. And your ball of cells is going to, I'm going to just draw the outline of it, sort of do this. So it starts to form this little cleft in here, okay? It's called a gastrula at this point. Those cells that line these certain little sections, like here and here around the outside, start to get orders that say, you guys need to become skin cells. You need to become the digestive system. You need to become heart cells. You need to become nervous cells or whatever. That's called specialization, all right? So cells will start to specialize at one point, differentiate, and become the cells that will do certain jobs. So you start off, that first cell, that first zygote, since that cell could become any cell, a nerve cell, heart cell, whatever, is called a stem cell. Then that cell divided, remember, and it divided into 2 initially. It became 2 new stem cells. They still have the ability to become any cell. And then eventually, they're going to become this ball of cells. Those are still all stem cells. Once they start to differentiate, so it's this last stage down here, once they start to differentiate, they start on a pathway. You can see these arrows right here, to become a certain type of cell, okay? That's a differentiation pathway. But one stem cell can either divide and become 2 stem cells, or it can divide to become a stem cell and a potentially differentiated cell, all right? Those are called progenitor cells. So a stem cell still has the ability to become any type of cell you need it to be. A progenitor cell is on its road to differentiation. It's committed to a pathway that says it's going to become this type of cell, a blood cell, a bone cell, a nerve cell, whatever it is. It has a pathway that's already determined. Okay, that's called a progenitor cell. Still isn't that neuron yet. It isn't that blood cell yet. It is a cell that is going to become, as it divides more, become those cells. All right. So stem cells have the ability to become any cell. Progenitor cell is a cell that is on its differentiation pathway. All right. Some terms to get to know. A stem cell is totipotent because this describes a cell that becomes anything. So stem cells have the ability to become anything. So their adjective that describes them is totipotent. They haven't differentiated yet. So remember my zygote I drew over here? Here's that ball of cells called a blastula or blastocyst. Here is this beginning. There's that little, where I was talking about the gastrulation here. You can see different colors in this right here, this gastrula that shows different cells in different regions, and they're going to start becoming different types of cells. So this group of cells right here will become part of the ectoderm. This group of cells that lines it in the light purple are going to become what we call the germ cells, or the reproductive line. These cells around the outside, what we call the mesoderm, which includes tissues. You're going to learn a lot of this later in next semester, but cardiac cells, skeletal muscle, red blood cells, and so on here. And then this little group down here in that little section is going to be called the endoderm, which is interior types of organs and stuff like lung, thyroid, pancreas, that kind of stuff. All right. So that's our differentiation. When it's here and here, they're stem cells, and they're not differentiated yet. They are totipotent. When it gets to this stage, we have moved past that totipotency. Some of them stay that way. Some of them stay stem cells because you still have to build new cells. But these that will continue on this pathway, this pathway, this pathway, or this pathway are starting to become specialized. They're differentiating. All right. They haven't differentiated completely yet, but they're getting there. We refer to them as pluripotent. So now they're on a pathway. And if I go back again, let me show you back again. See this pathway? This pathway doesn't say, hey, you will be a skin cell. This pathway says you will be a skin cell or a neuron or a pigment cell, right? So that is a pathway that says I could really branch into these three directions. This is the pluripotent pathway. This is the specialized pathway. So we haven't got to this point yet where it's completely specialized. We're on the pathway. But if it's on this pathway going this way, it can't do this, this, or this, right? So it can be any one of those three cells in that picture, but it can't become these, right? That's pluripotency. Totipotent, it could be any of those. It could be all of these things. That's totipotent. Pluripotent says it can be a limited number because it's on a certain pathway to become that group. All right, let's go ahead and get rid of all these. Okay, so it's on a pathway. It can be a restricted number or types of cells. So here's my stem cell. When it divides, it can become another stem cell. So it can become two stem cells, right? Or it could become a stem cell again and then go this way. So it's going to divide into two either way, but it can become this. Progenitor cell is that cell that's going on a pathway. So this is a progenitor cell going this direction, and this is actually 1, 2, because now it's differentiating. But this is another pathway. So these cells here can only become these group. These cells can be this. Notice they haven't specialized. This cell is not a neuron yet. It is on the pathway to become any of these group. That's progenitor. All right, a differentiated cell is a specialized cell, okay? So when things differentiate, they get a job. So once they get to this progenitor stage, again, they can only become this limited type. Once they get to a specialized stage, then they can only make more cells of themselves. And some don't even do that, like neurons don't make more copies of themselves. Red blood cells don't, but cells in the bone marrow will make more red blood cells. And then neurons you're kind of born with, and you're stuck with them the rest of your life. But now that is the only thing they can become, basically. So they're committed to a certain type of cell. And remember, that's pluripotency. Only a limited number of cells can be formed from that progenitor cell. So again, looking at this, here's the sperm and the egg. When they come together, we form a fertilized egg, also known as a zygote. Still stem cells. These cells are, a stem cell here, and then it will become a ball of stem cells. So as it starts to divide, it still is a division to stem cells. But then we start down this pathway, and that's when you see the progenitor. So this progenitor cell could actually form either of these two pathways. Once you get to this progenitor, it's a little more specialized. It can only go these two pathways here. It cannot go back and go this way again. And this one can only follow that direction. So that's that specialization. These are pluripotent. These are totipotent. Okay? That's an adjective that describes them. These are differentiated or specialized. So they cannot go back again. They can't go back and become something, go down a different pathway. That's not allowed in nature. However, not in nature, we have figured out how to take something like this skin cell, send it back to a stem cell state, and then make it go down a different pathway. Every single cell in your body has the exact same chromosomes. It just doesn't express all those chromosomes. So like my skin cell right here, My skin cell has a job of protecting, of modifying the heat in your body and that kind of stuff, whatever its job is, right? So even though it has every single chromosome that your neuron has too, they're exactly identical genetically, it expresses a different set of those chromosomes. It only uses the chromosomes that's going to help it do its job, right? Whereas the same thing here with the neuron. The neuron has all of the skin cell chromosomes. They all do. I mean, every single cell has all the same chromosomes, but it's shut down those chromosomes that tell it to do skin cell stuff, and it only uses the chromosomes that say, hey, be a neuron and do a neuron stuff. Well, what if we just reactivate those chromosomes that were shut down? So all these chromosomes in the skin cell for neurons, for bone cells, for whatever else, they're present, they're just not active. So what we've done in the lab is we've reactivated them, sent the skin cell back to a stem cell state, and then we can tell it, hey, why don't you become a spinal cord? And then maybe we could repair a spinal cord injury. Why don't you go down the pathway and become a pancreas cell? So have the skin cell become a pancreatic cell and make insulin where somebody whose pancreatic cells aren't making insulin properly, a diabetic, needs to have insulin injections. Let's just get some of the skin cells to take up the slack. Let's send those chromosomes back to a stem cell state, reactivate them, and make the skin cells do that job. That's a potential cure for diabetes versus a treatment for diabetes, if you can get other cells to do that. So we have been able to send cells backwards up that pathway in the lab. And we've had success, by the way, with this in lab animals like rats. We've been able to take them and send their cells backwards and have them produce insulin in a different cell than the pancreatic cell. Cured their diabetic disease. And that's what we would call therapeutic stem cells. Not the stem cells just to make a human being or a dog or a cat or a plant or whatever, but stem cells that we're going to use for a purpose of curing a disease or an injury. That would be therapeutic, okay? And there's different ways to get them. You can get one from a donor, so you would have to find somebody who has stem cells that are like a match, like you would an organ donor, right? We all, as adults, still have stem cells in our bone marrow. So you just take a bone marrow, get a bone marrow match from somebody, take some of their bone marrow, and replace it in that person. And we do that with leukemia. So give them some new stem cells from the bone marrow, and that person now will make healthy cells because they have somebody else's bone marrow in there. Umbilical cord is the same thing. So when a baby's born, the umbilical cord blood actually is stem cells too. You can harvest that, by the way. There's companies that will allow you to harvest the umbilical cord and freeze it. And so if that child ever needs, that would be self, by the way. But you can use it for another child, or you can use it for you take your own child's umbilical cord, you freeze it. If your child ever has a disorder and we have the capability of using the stem cells to repair that disorder, you could use that umbilical cord stem cells to reprogram them and potentially fix that. Bone marrow, again, you can, your own bone marrow. We could reprogram cells, and that's what I was talking about on the previous page. So that's taking something like a skin cell that is already highly differentiated, but sending it back to a stem cell stage against what it would normally do, and then tell it to go forward into something else like pancreatic cells, right? And actually have working pancreatic chromosomes that would produce the right enzymes. They're also working on it for muscular dystrophy. Muscular dystrophy is a problem with the protein that's being made, and the muscles deteriorate over time. So, if we can take stem cells or some sort of abnormal cells, send them back to stem cell phase, and then reprogram them to correctly produce that dystrophin, they could have actually healthy muscle cells. All of our cells also have this ability to kill themselves. If they're old, you don't want them hanging around. Get rid of them, replace them with a newer, younger version, right? If they're damaged, you don't want them hanging around. You definitely don't want them going through mitosis and making more copies of a damaged cell. So send them through cellular death. This is called apoptosis. Apoptosis is the term that means cellular death, and it's programmed and it's normal to happen. It's an orderly destruction of cells that are no longer doing what they're supposed to do. So if I want to get rid of a cell, here's my normal cell, but it's not needed anymore for whatever reason. Maybe it's damaged, maybe it's cancerous, maybe it's just not needed anymore. There are cells sometimes you build and then you have to go back and get rid of them, right? So maybe it's just not needed anymore. We're going to have to go through a set of steps to destroy the cell. So first thing, shrink the cell up, sort of condense it, start getting rid of things. Cut the chromosomes up, slice and dice the chromosomes, start cutting the organelles up, especially the mitochondria that has energy for the cell. And then the cell is going to start blebbing. Blebbing is this right here where it's bulging in sections. That means it's pinching off and it's going to start breaking up into pieces. So in apoptosis, the cell will eventually bleb. Two great words right there. Breaks off, breaks into little pieces, and then we can recycle all those little pieces and make new cells from those pieces. So it is a normal and healthy thing to do with your cells, right? We don't want to say, hey, it's death. It's death for purposeful reasons, all right? We do this during fetal development. We go through development of structures, a lot of development of structures, and very, very quickly, you can see the amniotic sac around the fetus here. You can see the development of the head, the arms, the heart in here, all of that. And you can look at skull and spinal cord and everything in there too. Here's the umbilical cord. We build structures and then we get rid of structures. All of you at one point, and you can't see it here, but all of you had a little tail. So very early on, before your mom even knew she was pregnant with you, you had a tail. So your DNA says build this tail. But as adult human terrestrial beings, we really don't need tails. So you go through the process of building it, and then you go through the process of destroying it, apoptosis, destroy all those cells, and what we're left with is just the tail bone. You can also see the structure of the hand right here. Look at that little hand on that early fetal development at 6 weeks. The fingers are not separated into fingers, they're all webbed together. So as we build the hand, we actually have webs that are connecting all that. But as that fetus develops further down here at 15 weeks, you can see that the fingers are dividing. Those cells that were there in that webbing were built, and now they have to be destroyed. So those cells are going through an orderly destruction so that your fingers can be separated. And you can have, on each hand, 5 fingers, 5 toes too. All right. Sometimes that goes wrong. You don't get a complete division. So here, this little child has what we call syndactyly, where that tissue never divided. That's easily taken care of. As long as it's just skin tissue, you just divide it and go through surgery. If it involves more of the bones and stuff, it's a little more complicated of surgery. But if it's just the soft tissue, it's easier to do. You can see syndactyly of the toes down here too. So, it's supposed to be there for reasons. It's also there for destruction of old and damaged cells. So sometimes we have to just get rid of cells that we don't want anymore, we don't need anymore, because they're not doing their function. Let's get rid of them and get new cells. At any rate, whatever the purpose is, I'm going to have to destroy the cell. So we have these enzymes that come in and start to break necessary components of the cells down so the cell can't do its job anymore. So get rid of the DNA. Slice and dice the DNA. Don't let it replicate anymore. That would be mitosis. You're just building more cells. We don't want it to do that. Cut that DNA and shred it up. So cut it up, right? Dismantle the cytoskeleton. Remember, the cytoskeleton is kind of giving the structure and form to the cell. And if you dismantle that, it's going to start pulling in, it's going to shrink up, and it's going to start blebbing at this point. So stop supporting the cell membrane and stop supporting the nucleus. Cut off its energy source. So break up the mitochondria. If a cell doesn't have a supply of ATP, it dies within 3 seconds. So without any ATP energy, the cell is going to die. So here's our main source of energy. Get rid of that, and so kill off that cell so that you can dismantle it. Abolish its ability to adhere to other cells. In other words, I've got a cell and it's in contact with other cells. If I'm going to kill this one off, I don't want it being attached to this one and this one. So you cut off its ability, and it's going to be able to break off, right, and break into parts. And then this stands for transport. Transport phospholipids. Remember, that makes the cell membrane and the organelle membranes and the nuclear membrane and everything. So we want to take certain phospholipids and go to the cell surface and attract white blood cells named phagocytes. That means big eater or cell eater. Phagocytes that come in and break down and just actually get rid of all of the cellular debris. So break that cell apart and then take those cell parts away. And that's it. We're done with chapter 3. And we'll see you again in chapter 4.